Hello, everyone. Welcome to our last webinar of this advanced bioinformatics series. So today we have our speaker, Chun-Han Young. He's a research fellow at the National University of Singapore. And today he'll be talking about MapBatch, so batch normalization with deep learning. And his talk will cover autoencoders for batch normalization, benchmark of MapBatch with other tools, and also present a case study of a successful identification of rare cell populations. So the talk will last around 45 minutes and will be followed with a Q&A session at the end. So if you have a question, please either raise your hand at the end of the talk, or you can write your question in the chat and I will ask Dr. Young at the end. So over to you.
Okay, thank you. Good afternoon, everyone. And I believe some in the audience will be from different time zones. So good morning or good evening to you too. My name is Chun-Han. I'm from the National University of Singapore, Department of Computer Science. So in this presentation, I will describe MapBatch, which is our method for batch normalization of single-cell RNA sequencing data. Okay, here's what I will cover today. First, I will start with some background and motivation. So many diseases involve rare cell populations. These are cell populations present in, firstly, very small numbers, and secondly, can only be found in subsets of patients. For example, only in certain disease subtypes or certain disease stages, or maybe only before or after some specific treatment. Some examples of these cells are, say, cancer stem cells and certain immune cell subsets. Now, single-cell data can potentially uncover such rare cell populations, but this is dependent on the data processing. In particular, on batch normalization, which is used to integrate data from multiple experiments together. So there are many popular methods for batch normalization. Some of the popular ones are like Surat and Harmony, and they perform very well in mixing batches together. However, what we found was that these methods could obscure certain cell populations. Cell populations in only a subset of samples and small cell populations were frequently merged into other cell types. So here's an example. This rare cell population in light blue can be seen when looking at a single sample. But once we integrate multiple samples together with batch normalization, this cell population becomes merged into other cell types. And in fact, on bioinformatics forums online, you can see that this issue is also encountered by other researchers. So there is a need for more conservative batch normalization, which can retain the biological signal and allow these rare populations to be found. So we designed MapBatch, which performs conservative batch normalization, just enough normalization to enable downstream analysis while trying to maintain as much biological signal as possible. In fact, for many downstream analysis, it doesn't matter much if some batch effect remains. So you actually don't need to go all out in eliminating the batch effect. So how does MapBatch achieve this? MapBatch uses autoencoders, or AE for short. An autoencoder is a neural network. It looks like this. It is frequently used in tasks like image recognition. The autoencoder can transform or project high-dimensional data. So over here, pixel-level data of these images down into lower dimensional space. So how it does this is essentially it uses lots of training data to learn the weights of the network that can encode this high-dimensional data into low-dimensional features, and then decode the low-dimensional features back to high-dimensional data, while minimizing the difference between the reconstructed output and the input. So once it has achieved this, the trained network is able to project the high-dimensional data into the low-dimensional space. And we know it can do this because it is able to reconstruct back the original input from the low-dimensional space. And this low-dimensional space represents the latent structure in these images. MapBatch uses autoencoders to project single-cell data into batch-free biological subspaces. But before I explain how this is done, let me describe our autoencoder architecture first. So the high-dimensional input is the single-cell gene expression data on the left. And this data is projected to a lower dimensional space encoded by the latent variables in the middle. And this lower dimensional space represents the underlying gene expression structure in the data. The lower dimensional data is then reverse transformed back to the original high dimension at the output layer on the right. Here's a little bit more technical details. Our architecture also incorporates a feature extraction layer. It extracts features in the form of known biological modules from the CAC database. It has multiple channels of feature extraction, and it performs regularization during training with noise and dropout layers. Okay, now I will explain how MapBatch actually uses the autoencoders. In MapBatch, an autoencoder is trained on a single batch of data, as illustrated here. So this is a visualization of the cells in gene space. This batch has 3 cell populations: B cells, NK cells, T cells. Okay. This data is used to train the autoencoder. The autoencoder learns to project the data into a lower dimensional gene expression structure, which is the biological subspace of this data. So we call this S, represented by this blue curve. When the data is projected onto S, it looks like this. And unsurprisingly, the 3 cell populations, B, NK, and T cells, are still well separated in S. And of course, this data can be reverse transformed to the output layer back into gene space. Now remember, this is just one single batch of data used for training. Now say we have multiple batches, batch 1 and 2. So batch 1 is highlighted in the blue shade and batch 2 in the green shade. Each batch has 3 cell types, once again, B cells, T cells, B cells, NK cells, and T cells. And notice that batch 2 is offset from batch 1. So this represents the batch effect between the 2 batches. Once again, the autoencoder is trained using just one batch. So it's trained using just batch 1. It learns the projection into S, which is the biological subspace of batch 1. Now, when batch 2 is projected onto S, this removes the gene expression signal orthogonal to S, while it retains the signal lying along S. So what does this do? Note that S spans the underlying gene expression structure of the training batch, but S does not span batch-based differences because it was learned using only one batch. So this projection selectively removes batch effect between samples while preserving the biological signal. Now in the latent layer, all batches are projected into the same biological subspace of the training batch. And when the data is transformed back into gene space in the output layer, you can see what's happened. So over here, the gray cells are the training batch, batch 1, and the colored cells are from batch 2. And you can see that some batch effect remains. And this is because the batch effect that lies along the biological space is retained. But in fact, much of the batch effect is removed compared to the original data. So specifically, the batch effect that is orthogonal to the biological subspace S is removed. So the batch 2 cells become shifted down to where S lies. So what happens when, instead of training with just one single batch, we train with multiple batches? Over here, we train with batch 1 and batch 2. So batch 1 are the dots in gray, and batch 2 are the colored dots. So the autoencoder ends up learning the projection into all the training data into both batches. So this is the projection that it learns. And this means that the batch effect is learned as well. So that is why we always train with just one single batch of data. Now, if we can only train with one single batch, what if that one single batch does not contain all cell types? For example, here we train with batch 1, the gray dots. And batch 1 contains B, NK, and T cells. But the other sample, batch 2, contains additional NK precursor cells. And these cells are absent in the training batch. So the autoencoder does not learn the difference between NK precursor and NK cells. And the NK precursors will be projected onto the NK cells. So as you can see here in the latent layer and the output layer, the NK precursors become mixed into the NK cells, over here and over here. In other words, cell types that are absent in the training batch may become merged into other cell types. So now we have a problem. There may not be a single batch containing all the cell types we may encounter in the study. And yet, we cannot train with multiple batches because then the batch effect will be learned. So, how do we cover a diversity of cell types during training? Our solution is to use an ensemble of autoencoders. The ensemble contains multiple autoencoders, and each autoencoder is still trained on a single batch. So here, autoencoder 1 is trained with batch 1, autoencoder 2 trained with batch 2, and so on. And since each autoencoder is trained with a single batch effect is not learned. But the multiple autoencoders in the ensemble still allows multiple training batches so that diverse cell types can be covered during training. But note that we do not need to train with all batches in our data set. We just need to select a representative subset of batches that can cover the different cell types. So after training, the autoencoders can transform the input data, and the outputs can simply be concatenated for downstream analysis. Okay, so putting everything together, here is an overview of MapBatch. First, we have an ensemble of K autoencoders. Each is trained with a single batch. Then the input data, which consists of N batches, is fed into the trained autoencoders. The batch normalized data in low-dimensional latent space are extracted from the latent variables to give you these matrices. And this can be used for latent level analysis, such as visualization, clustering, and so on. Batch normalized data in gene space can be extracted from the autoencoder's output layers. And these can be used for gene level analysis, such as differential expression. Okay, so that was a quick overview of MapBatch. So now let's look at some performance benchmarks. So we generated a data set with simulated rare cell populations. This is based on 7 batches of PBMC data from 10x Genomics. So we named these batch 1, batch 2, and so on up to batch 7. They were generated using different chemistries, including chromium V2, V3, chromium NextGen, some of them with and some without protein antibodies. So these differences can contribute further to the batch effects between the batches. Then we modified this data to add the rare cell populations. So what we did was, in batches 1 and 2, we perturbed a small number of CD20B cells to generate a population of perturbed CD20B cells. And this is a very small number of cells comprising only 0.5% of all the cells in the batch. We named these batches batch 1B and batch 2B, B indicating that it has the perturbed B cells. Similarly, we perturbed T cells in batch 4T and batch 6T, and we perturbed monocytes in batch 3 mono, 5 mono, and 7 mono. So a side note, to perturb a cell type, we selected 40 genes to upregulate and 40 genes to downregulate. So in the resulting data set, all batches contain the usual PBMC cells, while the perturbed cell types are contained in only subsets of batches. So the perturbed B cells contained in only batch 1B and batch 2B. Perturbed T cells are only in batch 4T and batch 6T, and so on. Okay, so here's a UMAP visualization of the cells without batch normalization. On the left, the cells are colored by cell type, and on the right, the cells are colored by batch. And you can see that the batch effect dominates. So for example, among the T and K lymphocytes, which are over here, batch 1 is located here. It's colored blue on the right. Batch 4 is over here in brown. Batch 2 is over here in yellow, and so on. Likewise, with the monocytes, batch 1 is here, batch 4 is here, and so on. The arrows over here indicate the perturbed cells. So the perturbed cells also cluster by batches. For example, for the perturbed T cells, batch 4 is over here, and batch 6 is over here. And for the perturbed B cells, batch 1 is here, and batch 2 is here. So this kind of strong batch effect makes downstream analysis very difficult. So we performed batch normalization with MapBatch. We used an ensemble of 3 autoencoders. Each autoencoder is trained with a single batch. So autoencoder 1 is trained with batch 1B, autoencoder 2 trained with batch 4T, autoencoder 3 trained with batch 7 mono. So these 3 training batches together can cover all cell types. So after normalization with MapBatch, the cells now group very nicely by cell type. So B cells are here, monocytes are here, T and NK cells are here. And the batches actually mix pretty well. So for example, among the B cells, all the batches are mixed pretty well. Likewise with the other cell types. Furthermore, each of the perturbed populations now form its own cluster next to its parent cells. So the perturbed B cells are over here, located next to the parent B cells. Perturbed T cells are here next to the parent T cells. And the perturbed monocytes are here next to the parent monocytes. So in other words, MapBatch can maintain these rare cell populations while still effectively integrating the batches together. So as a side note, note that the 3 training batches together can cover all the cell types, right? But if we only train with batch 1B, so that the training data only sees the perturbed B cells, then only the perturbed B cells can be distinguished. The perturbed monocytes and T cells will be absent in training, so they end up being merged into their parent cells. So with MapBatch, it is actually very predictable what happens when a cell type is not represented in training. Okay, so we tried 3 other methods: Surratt, Harmony, and Liger. So these are some of the more popular methods for batch normalization. However, these methods were not so successful. So for example, on the left, the results for Surratt. So with Surratt, the perturbed monocytes and the perturbed B cells were mixed into their parent cells. Only the perturbed T cells could be distinguished. And the reason for this is, so even though we specified 3 batches for Surratt to use as its reference, Surratt will use only 1 batch as its base reference. Okay, and Surratt chose batch 4T as its base reference. So batch 4T contains the perturbed T cells. And when the other batches are integrated into this base reference, the other perturbed cells will align with their parent cells. So that's why only perturbed T cells can be distinguished, while the other perturbed cells are mixed into their parents. So in the middle is the results for Harmony. So with Harmony, the perturbed B cells were mixed into the parent cells. The perturbed T cells were actually still split by batches. These are the 2 batches of the perturbed T cells, and one of them ended up near the monocytes instead. But Harmony was able to distinguish the perturbed monocytes. With Liger, the perturbed monocytes and B cells were mixed into their parent cells. And the perturbed T cells actually ended up next to the B cells, next to the wrong cells. So with these other methods, they cannot deal with cell types that are present in only a subset of samples. They either overcorrect and merge them to the next nearest cell types, or even worse, they may align them to totally different cell types. Whereas MapBatch successfully delineated all the 3 perturbed subpopulations. All right, next, we tried using the batch normalized gene level outputs for differential expression analysis. So note that MapBatch and Surratt can produce gene level outputs, batch normalized gene level outputs, whereas Harmony and Liger do not. So we did differential expression between each perturbed cell type and its parent cells. So for example, we did differential expression between the perturbed B cells and its parent B cells. And we wanted to see whether we can predict the genes that we upregulated or downregulated to create the perturbed population. Okay, so we want to predict the up or downregulated genes in the perturbed cell type. The left chart at the bottom shows the results for the MapBatch processed data. So this is an ROC chart. So essentially, a higher curve indicates better prediction performance. And there are 3 curves for the 3 perturbed cell types. So over here, we can actually predict the genes in all 3 perturbed cell types very well. Now on the right shows the results for Surratt, and only the genes in the perturbed T cells could be predicted, but not the genes for the perturbed monocytes and B cells. And actually, this is not surprising, because in the previous slide, you saw that the perturbed monocytes and perturbed B cells were merged into their parent cells. So actually, the gene expression differences were actually removed by Surratt. And finally, let's look at the running time of MapBatch. So MapBatch's running time scales linearly with the number of training batches, not the number of batches to normalize. Okay, so each training batch takes about 2 minutes to train the autoencoder. So this line at the bottom is the running time with 1 training batch. Above that is a running time for 2 training batches, and then a running time for 3 training batches, around 2 minutes per training batch. Typically, only a small number of training batches are used, say 2 to 4 training batches. So you could expect your running time to be about 4 to 8 minutes. After training, performing the actual normalizing takes negligible time. So it takes just about 3 seconds per batch. So you can normalize huge numbers of batches and the running time doesn't increase by much. So in other words, MapBatch is extremely scalable to process large numbers of batches. In contrast, scRNA's running time scales linearly with the number of batches to normalize. So it takes a lot of time to process a large number of batches. Over here, I did not show the running time of Harmony or Liger. However, both are also faster than scRNA, especially Harmony. Harmony actually scales very well with large numbers of batches. Okay, now for the last part of my talk, I will show a use case for some real-world data. So we used MapBatch to process a cohort of multiple myeloma samples. So we have 14 samples and 2 healthy controls. The UMaps here are colored by cell type. The UMap on the left shows the cells before batch normalization. And over here, you can see that the batch effect dominates. Each cell type, for example, the plasma cells, is shattered into many small clusters corresponding to the different batches. So some sort of batch normalization has to be done if you want to analyze this data. So we use MapBatch to normalize the data. And after normalization, now the cells group very nicely by cell type. And within each cell type, there is very good batch mixing. We then did unsupervised clustering, and we identified 20 clusters. The chart on the right shows the number of cells in each cluster. And it turns out that there were 3 small clusters that looked very interesting. So these are clusters 16, 18, and 19. So these could not be reliably annotated. Okay, so we annotated each cluster using a software package called MapCell. So many of these clusters could be reliably annotated by MapCell, except for these 3 interesting clusters. These are rare cell populations. They comprise less than 1% of the cells, and they are found in only subsets of patients. So these are so-called cryptic cell populations, and they may be interesting, and they may have biological significance. So it's important to be able to find them. So to check that these rare cell populations are real, we visualize the individual samples using their uncorrected gene expression data. So this data was normalized only for library size and then log scale, with no further batch normalization done. This way, we can see the cells in each sample, and it's untainted by either batch effect or any batch processing. And we can observe that these rare cell populations are present as distinct clusters in individual samples. For example, in sample N201, cells from cluster 16, 18, and 19 can be seen, and they form very obvious clusters separate from the other cells. In this second sample, you can see the cells from cluster 16 and 18, and so on. In this cluster, in the last sample, this sample only has cells from cluster 19. which are over here. So these cell populations appear to be real, and they are not technical artifacts driven by either batch effect or MapBatch itself. However, when we tried other batch normalization methods, we tried Seurat, Harmony, Liger, and SCVI, some of these populations were obscured and they could not be identified, whereas MapBatch could maintain the biological signal to identify these rare populations. Okay, so in conclusion, batch normalization involves a trade-off, right, between removing batch effect and retaining biological signal. And most popular methods out there actually lean towards batch effect removal. This is at the expense of losing biological signal. In contrast, MapBatch performs conservative batch normalization, and it can retain more biological signal for downstream analysis. And specifically, with this higher biological signal, it enables the discovery of rare cell populations. So how MapBatch works is it uses an ensemble of autoencoders. Each autoencoder is trained with a single batch, and it learns a projection into the batch-free biological subspace of that training batch. And an ensemble of multiple autoencoders allows learning a diversity of cell types while not learning batch effect. Currently, MapBatch is available as a Python package. It comes with tutorials and guides for the entire processing pipeline, from data pre-processing to model training, model selection, and batch normalization, and even some downstream analysis. This is not yet published online. We are currently working on a paper, but if you want to try out the package, you can feel free to email me. Okay, so thank you for your attention. This work was a collaboration between NUS, AI Singapore, and the folks at Singleron. For more information, you may contact us at these addresses. Okay, thank you.
Thank you. That was a really interesting talk. We actually have quite some questions, so I'll just start straight away. So the first question is, how many cells of a rare cell type are minimum required for MapBatch AE to identify them as a rare population?
So typically we select 2,000 cells. Where's my... Yeah. For most of my experiments, I just take 2,000 genes. Wait, sorry. Was the question number of genes or number of cells?
Number of rare cell types.
So the size of rare cell types, it can be very small number of cells. So for example, in some of the data sets I worked with, we found some small populations consisting of like 20 cells. And these look real, and MapBatch managed to basically identify them. So a tiny number of cells can be found.
Okay. And second question, how does technical noise such as ambient RNA or differences in sequencing depth bias the identification of cell types?
Yeah, so that is additional, those are additional sources of noise that you should try to deal with in the pre-processing step. So when I analyze a set of data, I'll typically run like SoupX to remove ambient RNA. And the other standard pre-processing steps are taken first before I run MapBatch.
Okay. Do you have a specific training data set you can use or publicly available data sets?
So of course, I have the synthetic data set, which I talked about. The other sets that I'm using, they are with ongoing research projects. So those data sets, of course, I cannot release with the MapBatch package. But the package is currently comes together with the PBMC data, which it's actually based on real data anyway. So I think it's quite a good data set to use to experiment with our package.
Okay. Another question is, does the algorithm come with an automated system for refining ROIs, or is it left to the technician's discretion to determine boundaries for cell segmentation?
Sorry, can you repeat the question?
Does the algorithm come with an automated system for defining ROIs, or is it left to the technician's discretion to determine boundaries for cell segmentation?
Sorry, what's the ROI? I'm not familiar with that term.
So it was written by Zato Sa. I don't know if I can allow you to talk and then you can. So ROI is region of interest.
Region of interest. I'm not familiar with that concept. Maybe he can elaborate what he means.
Okay, so I'll allow you to talk and then you can ask the question if you want to. You are muted, but you can talk if you would like. Okay, maybe we can come back to that question later. So I have another question. Does the percentage of cell type in each sample matter for the MapBatch?
In fact, no. So when we select, okay, so if we have a sample for training, we actually do some sampling of the cells in that sample to cover the different cell types in that sample, which are given to the autoencoder to learn. So this makes sure that even cell populations with a small number in that sample are sampled more deeply, so they are equally represented as the more common cell types in that sample. So this way, even rare cells in the sample are represented well during training. Okay. So MapBatch is able to pick out these rare cell populations.
Okay. Another question is, how important is it to have the KEGG modules in the MapBatch, and can it be replaced by other modules like reactor modules or protein-protein interactions?
Yeah, for sure. So I did not fully test, what's the performance benefits of using KEGG modules or not. But I suspect, given what we know with autoencoder training and so on, other biological modules that make sense will probably work just as well.
Okay. So another question is, can the genes be extended from gene 1, gene 2 to more genes, or is there any recommended number of genes to process in MapBatch?
Okay, so yeah, when I put gene 1, gene 2 here, of course, I don't mean that we can only work with 2 genes. So this is just a visualization of the cells in some sort of gene space. Typically, I'll use 2,000 genes in my architecture. And the number 2,000 is also pretty arbitrary. 2,000 because that's the number that the standard seurat pipeline uses. So, you could use more genes or fewer genes if you wish. And how I select these 2,000 genes is similar to seurat, getting the most variable genes in the data set.
Okay. Is it a must for cell types to be pre-imputed to run MapBatch?
No. That'd be necessary. No. We do some sort of clustering to select the training samples. So we want to select training samples that cover a diversity of cell types. And how we do that is, we do clustering on each sample, and we select the samples that have a large number of clusters. So this is like a proxy for the diversity of that sample. Of course, the cell type of these clusters don't need to be known beforehand. So no, cell types don't need to be pre-imputed.
Okay. So going back to the question before, they have clarified, so cell segmentation is used to determine specific cell types within a heterogeneous cell population.
Okay.
So the question again, does the algorithm come with an automated system for defining region of interest, or is it left to the technician's discretion to determine boundaries for cell segmentation?
I would say that it's left to the technician's discretion. So for example, what I usually do is I will do clustering at a range of resolutions. And I will try to actually use pretty high resolution to get more clusters to begin with. And then as a curation step, I check that each cluster is real. And if it's not real, I will merge it with its nearby clusters. So, no, but of course, this is an optional step. If you have your own protocol for determining what fine level of cell types you want to analyze at, then you can use that, of course. MapBatch is just a method to basically get the batch process data, and then downstream analysis is up to you.
Okay. We had one raised hand during the talk from Kane To. I will allow you to talk now if you would like to ask your question. So you can talk if you would like. Okay, maybe maybe they're not there anymore. I think we have you have answered all the questions that we have had. So if there are no more questions, I would like to thank you again for the really informative webinar. So this talk will be uploaded onto our YouTube channel, the Single One YouTube channel. So if you would like to see it again, you can go back and visit our YouTube channel. Okay, so thank you, everyone, and I will see you soon.
Okay, thank you.
Bye.
Bye.